Pointer. Okay, thanks very much, Sarah. It's an honor, and I'm very appreciative to the SID and to the program committee for this great honor, not only for the breadth of the contributions that we in Montagna made to the society, but also for the previous winners and the ongoing amazing contributions they're making to our society who have won this award. Now, William Montagna was a bird watcher, and he quickly understood that watching birds and watching skin could provide very unique and profound insights into biology and medicine just by watching. And what I'd like to do today is to tell you, over the last few years, our Stanford groups from watching skin, insights into tumor evolution, autoimmunity, and regenerative medicine. So these are my disclosures. So today, initially, I want to talk about biological resistance. So biological systems, as you can see here, ensure survival by diversifying their genomes. And so they do that by changing epigenetically and genetically so they can evolve and maximize their reproduction in the microenvironment. The AIDS virus is a good example. We thought we had it cured by using protease inhibitors. We were wrong. We needed multiple combinations, various ways, to now actually make big dents into treating the AIDS epidemic. Similarly, in antibiotic resistance, we thought we were really great by using INH for drug-resistant tuberculosis. We were wrong. We needed multiple drugs, combination therapy from multiple different angles. What about cancer? What about cancer? Is it more like tuberculosis, or is there one single pathway? The problem of tumor resistance is that it remains one of the most challenging roadblocks to effective treatment. Tumors are heterogeneous. They create resistance that then help them to survive in multiple different microenvironments to keep dividing. For precision cancer therapy, we've heard throughout this meeting, we need to know the spectrum of resistance pathways at the start of diagnosis and then figure out which ones are available to the tumor and develop therapies for them. Example is trying to figure out how to leave San Francisco using a Google map. We need to figure out the rate at which they're leaving San Francisco. We also need to figure out all the different ways that they can leave San Francisco. So, for example, at the start of diagnosis, you might go down Highway 280 to get down to Palo Alto, but then as the tumor evolves, you really realize that you can actually go over the Golden Gate Bridge and the Bay Bridge together. These are the ways that tumors can evolve resistance mechanisms over time. Our job is to figure out how to block those pathways and kill the tumor. And so what we've done in our group is to use the basal cell carcinoma on the skin as a model for tumor evolution and resistance and combination therapy. And I was very lucky and fortunate, as Sarah mentioned, as a postdoc to be involved in a group which included Irvin Epstein, who's in this audience, as a postdoc in Matt Scott's lab, to really as a group of individuals to understand the connection between, the intimate connection between the hedgehog pathway, listed here, and human basal cell carcinoma, as well then over the last 15 years, about 25% of all human cancers involving the hedgehog pathway. And at the time we discovered that patch loss here in Gorlin syndrome resulted in activation of the smoothened G protein coupled receptor, which then leads to activation of this GLI transcription factor, which I'll talk about more, turn on genes and cause basal cell carcinomas in Gorlin's patients, which are genomic, genetically inherited, or sporadic basal cell carcinoma, which we get from sun damage. And then we and others, such as Ange Dlugos and others in our society, have then spent the last 15 years understanding this pathway and its intimate connection with basal cell carcinomas. I was also fortunate to then show at Stanford with Paul Kavari, who then developed a human regeneration model to model human disease and cancer, to show that then hedgehog pathway was sufficient to actually induce basal cell carcinomas in a human system as well. Then over the years, again, being fortunate to work with a group of very talented clinician translation and translational physician scientists at Stanford, including Ann Chang, who's in the audience, and Jean Tang, and then more recently, Kavita Sarin, to really understand and work with the industry to take the first hedgehog pathway inhibitors targeting smoothen, the smoothen receptor, to then look and show how effective they can be based on the genetics that we showed earlier as a postdoc. And you can see that beautifully these non-surgically resected basal cell carcinomas, using inhibitors to smoothen, massively can shrink the tumor and be And that led to approval 4 years ago of the first hedgehog pathway inhibitor, which now many others are available and are being tested. So this was the answer that smooth inhibition led to the eradication. Almost surgeons in the audience, and I wouldn't be talking to you today. Because tumors evolve. And this is an example of the sneaky ways that they develop new ways, such as new ways of leaving San Francisco, new freeways to get out. And that is using, and this was actually first observed by Yan Chiang, where you can see that the tumor is there, it goes away, and then approximately 7 months later, individual clones of resistant tumors appear in the skin. And so this, again, is like bird watching. You can see the skin. You can notice that, and I'm friends with Yan Chiang, notice that the individual clones of basal cell carcinomas would appear. So as a geneticist, and we know that in subsequent RNA-seq studies that these are all hedgehog dependent. So if you're a basal cell carcinoma, you're completely addicted to the hedgehog pathway. As a geneticist, this is amazing because this says that if you watch these tumors and biopsy them and do genomics on them, this is like doing a massive second-site suppressor screen for the hedgehog pathway in humans. And so you can see anything that upregulates the GLE transcription factor activity is going to develop resistance. So that if you then take your patients, biopsy many of these different tumors, do genomics, and understand what ways tumors evolve with time. It's an amazing system to understand tumor evolution. And so the next few slides, I'm just going to go over some basic questions that the Stanford Basal Cell Carcinoma Consortium has really addressed. And that is, why do some basal cell carcinomas develop resistance while others don't? How do basal cell carcinomas escape smooth inhibition? What pathways do they use? And can we drug these targets for rational combo therapy? Okay, so the first observation that our group made was that variable responsiveness of naive basal cell carcinomas to the smoothing inhibitors. It was the background, the microenvironment on which the driving oncogene, the hedgehog pathway, was induced, which led to the rapid or not so rapid change out into resistance. This is like if you're leaving San Francisco, you're driving slow or you're driving fast. What is it? And we noticed that in Gorlin syndrome patients, this is work from Gene Tang and Irvin Epstein, treating smoothing inhibitors with Gorlin's patients, that they respond, almost 100% of them respond to the smoothing inhibitor, but they have very low resistance. You can treat them for years. If they take them off the drug, the tumors resolve, they'll come back. You give them back the drug, the tumors go away again. They don't develop resistance at a very high rate. Sporadic patients, sporadic basal cell carcinomas, the small ones actually respond very well, but once they get large and heterogeneous, or on ultraviolet damaged skin, where there's a lot of mutations, the actual response is low, 40% at the time of diagnosis, and Chang showed that 20% per year will develop resistance over time. And then we, in our group, also identified a novel basal cell carcinoma type of where you have the sonic hedgehog translocation, and those respond very well as well. Why is this? Kavita Sarin has shown as well that basal cell carcinomas, sporadic ones that are big, have enormous numbers of single nucleotide variants. They have a high base pair mutation rate compared to other cancers, one of the highest that's known. In fact, our studies showed that 42 non-silent SNVs per megabase of DNA, which is very large. Another study showed about 75. Squames are about 33. In contrast, leukemias, 0.6. So why is that? And also, I'll just mention that Gorlin's patients have very low backgrounds of non-silent SNVs. So this difference is what we think is likely causing this enormous mutator phenotype on which the tumor was induced, and allows the evolution with time of the tumor. So Scott Atwood in the lab really wanted to take this to heart, and he said, okay, let's see if all these SNVs that we see are actually functional. And so he basically did this enormous thing where he took smooth invariants, for example, and transfected them into smooth and null cells to ask which ones are functional. And because these are recurrent non-silent SNVs, you'd expect all of them to be functional. But what Scott found was that some of them were actually gain-of-function mutations, but surprisingly, many of these mutations, which were recurrent in many different tumors that we see in our patients, were loss of function, and many were basically neutral. So this led to the idea that, in fact, what this mutator phenotype is doing is creating both loss of function, activating mutations, and neutral mutations in this realm. in order to try the best they can to actually develop resistance. This led to this model where we basically believe that the way that basal cell carcinomas evolved with time is by rolling the genetic dice. They're trying to just to mutate the heck out of the genome. Sometimes you lose, sometimes you win, but if you win, you basically win the lottery and you become immortal and can evolve with time. So they roll the genetic dice. In contrast, leukemias actually evolve by translocation to very low SNVs. And so this difference in tumor evolution is very clear in our, and we also see this in squamous cell carcinomas as well in the skin. Okay, how do basal cell carcinomas escape inhibition? The second, what are the ways they leave? So and also what mutations in the hedgehog pathway can teach us? So we noticed, for example, in smoothen that over half of the mutations are actually in smoothened. And what is this? And this is with very high coverage, so we have a very confident in terms of the sequence that is developed. What do we know about that? What can it learn? In fact, what we can see is that if you then map these mutations, these variants, onto the crystal structure developed by, for example, Ray Stevens, you can then learn a lot about the protein itself. Smoothen is a G protein coupled receptor. Here's the crystal structure, 7 transmembrane domains. And you can see that in the crystal structure that he developed for this is a very different smoothen inhibitor. This is how the ligand fits into smoothen receptor. What we showed is that actually then these are ligand binding mutations which prevent the ligand from binding, the inhibitor from binding, which allows it to then confer resistance. But actually it's very different than the one that was in the crystal structure. So if you do this with multiple different inhibitors in the treated patients, you can then develop what is the whole extent of the dynamic pocket that these inhibitors can actually use, and then use that to do computer-aided drug design to make a better inhibitor in the future. Moreover, you can see that there are many mutations that are not in the ligand binding pocket. These are actually constitutively active mutations that render smoothen active. There's been a controversy in our field about smoothen, whether it's a G protein coupled receptor or not. And in fact, these mutations lie in a region in the membrane which then confer autoinhibitory interactions between the various helices. If you then use this crystal structure and you turn this thing on its side, these 4 constitutively active mutations that we found form electron-dense ring in the middle of the membrane, which basically holds smoothen in its inactive state. And so that mutations in any of these different regions will actually then allow the smoothen receptor to become constitutively active. And in fact, if then you use this and then guess based on this electron density where this ring is, you can look at the different mutations and variants here, here, and here, and make a guess where other constitutively active mutations can be. And this can help you in terms of predicting in a personalized genomic way which are going to be constitutively active in resistance and which are not. Okay. So I've told you about why they develop resistance fast and about how basal cell carcinomas develop smoothen resistance. What are the escape pathways? What are the roads out for this tumor? And I want to tell you about 2 particular vignettes to give you an example of how these things work in terms of pathway switching, polarity pathway, and the tumor microenvironment and how important it is, because we've heard a lot about that at this meeting. So first, pathway switching. So we know that basal cell carcinomas are completely addicted to the hedgehog pathway. They have to turn on the Glee transcription factor to survive. Okay, so what we noticed in our group and had published, and Chang noticed, was that in the space where there was a basal cell carcinoma, a squamous cell carcinoma grew out. That was not what we expected. So if you look, very classic, well-differentiated basal cell carcinoma in the same field where the squamous cell carcinoma, where the basal cell carcinoma grew out. Many people in this room have contributed to the understanding that there are cancer stem cells in the skin that are bipotential. These cancer stem cells can make both squamous cell carcinoma and basal cell carcinoma, and a wide variety of mouse modeling, talking about mouse modeling of disease that Nicole talked about yesterday. You can then mouse model which, if you drive multiple different oncogenes in these cancer stem cells, what do you get? Well, what you get is if you drive hedgehog pathway in the cancer stem cell, in particular in the K14 or K15 positive progenitor cells, you get BCCs. If you drive RAS pathway or lose Notch in those same cells, you get squamous cell carcinomas. So then the observation at the time, again, the patient walks in, you see a squame coming out. you know the biology that other people have developed, and you can then see that the cell of origin gives you a squame for BCC, if with Notch and RAS, a BCC for hedgehog or a squame for Notch. If you then use a smoothing inhibitor that blocks this pathway, where do you think the tumor is going to go? If there's any type of RAS activity or RAS mutations in this background or any loss of Notch pathway, it's going to go to a squame. So it's not that surprising, knowing the wide variety of biology that people in this room have developed, that you then make this prediction that pathway switching is a very viable alternative to get out of San Francisco. You just take the Bay Bridge. Okay? And so, but in order to do that, you have to prove that these things are related by lineage. Okay? So mouse modelers do it by using Cre-Lox systems. Humans don't like it when you put Cre-Lox in their bodies. And so we tried to develop a novel lineage tracing system to really prove that this squame came from the BCC. And you can do that by whole exome sequencing. So if you take the normal blood, basal cell carcinoma that's sensitive, that came before, and then the subsequent invasive squame that came out that's resistant, and then do whole exome sequencing, you can actually compare the variants, because there are a lot of them, in between the different ones. And if the BCC and squame share very similar numbers of variants over the entire genome, like over 3,000 variants, then they're related by lineage. If they don't share any more than the normal blood, then they're just two metachronous lesions that have arise together. And in fact, what you can see is that in the basal cell and squame together shared over 91% allele frequency together, really supporting the idea that this invasive squame came from, and resistant squame came from the sensitive BCC in the same spot, resulting in and confirming the fact that pathway switching really is an alternative mechanism for resistance in our skin. And because our skin has so many mutations on UV background, this is really a viable alternative. And in fact, what you can see is that in the pretreated basal carcinoma, you see K14 up in GLE, and in the squamous cell carcinoma that's related by lineage, they basically turn all this off because of inhibition, and then turn on phospho-ERK and turn off GLE to become pathway switched. We published this paper recently in Cancer Research. Another group, Katie Ranselhoff and Kavita Saran, also found another patient which actually is more intermediate. So this patient here is actually a complete pathway switch. The patient that was published in the New England Journal, subsequently by our group as well, also showed an intermediate pathway switch. And I want to point out also that this paper in 2006 by Stu Jaspa's group, where he actually did the two-step squamous cell carcinoma model in mouse and then shut off the RAS pathway using the AFOS system, what he saw was that squames went down, and then they then developed sebaceous glands, showing the reverse pathway, that squames, when you block them, can actually go toward the hair follicle or basal cell carcinoma lineage. So the implications for this is that squamatization of epithelial cancers can then lead via inflammation, or accumulated mutations can actually lead to pathway switching, alternative driver oncogenes and drug resistance. And we see this in other types of cancer, such as non-small cell lung cancer, where lung adenocarcinomas can pathway switch into lung squames and back and forth. And this is really a viable option to think about when you're trying to think about combination therapy. Okay, so I just told you that the hedgehog pathway is critical. And so that if they don't pathway switch to squames, but they decide they want to stay in basal cell carcinomas, they have to figure out a way to turn on GLE. And what I would do is, using this amazing patients that we have, that have really shown us two new ways that really hadn't been discovered prior, using forward genetic screens in Drosophila and vertebrates, as ways to actually turn on GLE in the tumor situation. So previously, those were all they're all necessity screens in Drosophila, things that are required for the hedgehog pathway to work. In cancer, it's a sufficiency screen. It's basically what can happen to turn on GLE, and the cell uses any way it can to turn it on. What it turns out is that the things that we're discovering are present in the normal cell. They're just present as backup systems and at a low frequency and in niche systems to actually, but in the state of cancer, those come to the fore because they're the easiest way to evade the drug. And so the first really is polarity signaling. The hedgehog pathway is really known, and what the polarity system is, as shown by Scott Atwood, who's now has his own lab at UC Irvine, is is really a system which tells one part of the cell from the other part of the cell, from apical, from basal, from one cell division partner from the other, and really is a beautiful way in terms of cell fate decisions during development to induce cell fate. So this intimate tie between the hedgehog pathway and the APKC part 3, part 6 system, it's really a wonderful partnership. APKC regulates the actin cytoskeleton, endocytosis, and really, and as we know and we'll study celiogenesis, it's really now becoming an emerging oncogene which is now gaining much more fame and attention in the field. And Scott found this via a limited mass spec screen, which I don't have time to go into, but the work is published, but really shown that in smooth inhibitor-treated basal cell carcinomas, that this activated APKC kinase was highly upregulated and was a beautiful marker for resistant BCCs in our patient population. Scott went on to show that, developing an APKC inhibitor called PSI, which was very potent at blocking the activity of PSI, that it can actually beautifully inhibit the growth of resistant basal cell carcinomas in vitro, whereas the cyclopamine, which is the smooth inhibitor, could not do that. And in a mouse model, which was in collaboration with Jean Tang at Stanford, this PSI, blocking the APKC pathway in both sensitive and resistant tumors, can have a very nice effect in stunning the growth of these resistant BCCs. The aha was that because this kinase, which we found in our patients, actually phosphorylated GLI on the staining binding domain. This is a very unusual situation because in general, when you phosphorylate a transcription factor on its staining binding domain, it prevents it from binding DNA. But we noticed is that, and what Scott noticed was that the zinc finger protein actually binds in the DNA. So this is the DNA is in yellow, is in gray, and this is the helix. But part of the zinc finger in between in this regulatory region, in the non-binding part, actually sticks out above the DNA, much like RNA polymerase has tails and histone has tails that basically stick out above that can be modified and phosphorylated. And it's there that the APKC phosphorylates the zinc finger and activates it to allow it to bind to chromatin. And so I asked if you want to talk to Scott afterwards or myself about this, I can go into more detail. But really, this actually suggests that the entire conserved family of zinc finger proteins that regulate decisions in terms of development and differentiation all have this common property. And now it's leading to this much larger investigation into what these intergenic regions are doing and how to modify them, just like modifications on histones or RNA polymerase are well known. And indeed, if you then use PSI, you can then basically show that in doing chromatin immunoprecipitation, that normally GLI1 will bind to DNA. If you use this APKC inhibitor, it no longer can associate with chromatin. And then, even though it's resistant, it can't find the DNA and it can't develop. Okay. And so this has led to, again, an understanding and a whole area of a pathway which is upstream of PKC, which then leads to the phosphorylation and activation on chromatin of GLI. Okay. What about the tumor microenvironment? This is a very big part of the emerging understanding of tumors in our field. And we had a lot of talk by Roger Perlmutter on Thursday about immune checkpoint regulation. And I just wanted to point, and that's also a very important part of basal cell carcinoma development. And I just wanted to point out on this poster by An Chang, looking at PD-L1 and CD expression in correlating BCC response to hedgehog inhibitor this afternoon. But also a new pathway, which is identified by Ray Whitson, a talented postdoc in my lab, which is the serum response factor MRTF pathway in transducing tumor microenvironment signals to activate GLI. And this actually is an exciting pathway because, like APKC, it regulates the activity of GLI in an unexpected way, and also suggests that in an epigenetic way, rather than a mutational way, you can then get tumor resistance if you find the right microenvironment. And so Ray, I told you before that BCCs have a very high rate of mutagenesis. He used a mouse model, first generated by Jean Tang and Irvin Epstein, where then you develop resistance in allografts. This mouse actually has a very low rate of mutations, of SNV rates, very low in the hedgehog pathway. So these mice are more like the Gorlin syndrome patients that don't develop resistance. but they develop resistance. So how is that? And so what Ray noticed using multi-component analysis, and this is a very unique and wonderful way that I ask you should talk to Ray about afterwards, he identified the serum response factor as a highly active part in the resistant tumors. We also saw it in our in a subset of the human BCCs as well. What is the serum response factor? SRF is a transcription factor pathway which co-binds with a number of different partners to transduce the signal from the tumor microenvironment. These signals are inflammation, autoimmunity, and also a wide variety of microenvironment and vascular influences which actually lead to the activation of the serum response factor and to turn on activity. Its activity is regulated by nuclear cytoplasmic localization, so if you see it in the nucleus, you know it's active. And what Ray noticed was that SRF, well called SIRF actually forms a complex with GLI in these resistant BCCs. It chips to the GLI, with GLI to the GLI locus, and it can form a complex by immunoprecipitation. What we also noticed was that in mouse, in sensitive BCCs, SIRF is in the cytoplasm in its inactive state, but in the resistant tumors, you can see that it's driven into the nucleus and active on chromatin to turn these GLI transcription targets on. And also we see in a subset of our human tumors that don't have canonical hedgehog pathway mutations, which is about 25%, you can see that then these guys are also driven into the nucleus. Really affirming this pathway as a novel microenvironmental transduction pathway which allows these tumors, which have very low other S&V mutations, to survive, likely until they actually have enough survival time to get and become mutated. Ray wanted to show that one of the binding partners was the myocardial transcription factor, MRTF, which transduces actin cytoskeletal changes, which include actually mechanical transduction, and showed that in subsequent studies that if you use an inhibitor to MRTF, which is actually biologically available and very useful, might be very useful in the clinic, actually has beautiful inhibition of these resistant tumors. And so what I've shown you in this vignette is that you can use and watch human tumors and use novel biological techniques to really look at the various escape pathways that tumors use. I've shown you about pathway variants within the hedgehog pathway, such as smoothened, suppressor fused, and now Noel Mullen in my lab is actually looking at mutations in the cilia itself, which are very exciting. Kinase, which is the APKC, which will actually phosphorylate GLI and then allow it to bind to chromatin, and this SIRF MRTF tumor microenvironment transduction pathway, which once GLI is bound to DNA, you'll then actually activate it and ramp up its signal activity. And if it can't do that, it gives up and basically pathway switches to another kind of cancer. So these different pathways really summarize at least what we know about today. There's actually about 3 or 4 more that we're working on in our lab, but I think will hopefully round out the ability to translate into therapy. So what does this mean? With this talented group of the Stanford Basal Carcinoma Consortium, we then can use this basic science knowledge, animal modeling, chemical biology, to then develop phase 0 biomarker studies in our patient groups at Stanford with this talented group of translational and clinical biologists, many of which are here at this meeting, to then basically develop, and these are some of the trials that we're doing at Stanford to really look at combination therapies and see how best to treat these basically refractory tumors in our patients. So in the last just couple minutes, I wanted just to remind you that you can look at cancer and see how it works. But what about other things? For the trainees in the audience, look closely at the skin, and then you can really understand and make profound influences into biology. For example, the hair. The hair is a beautiful system. You can see it. It's wonderful. But really, it's a beautiful stem cell system. What's not appreciated is that the hair stem cells lie in a microenvironment. They have to live in this microenvironments here, and the hair is a beautiful biosensor for a wide variety of developmental signals. And we know from a lot of work that we've done in our lab and others in the room that there's activation signals and quiescence signals that really balance the growth and not growth of hair. And so for disease like, for example, and then Angela Cristiano, for example, has shown that JAK-STAT inhibitors are actually normal environmental signals that actually cause hair cycle arrest in telogen. and work from George Cottrell's group, and actually beautiful presentations yesterday, show that prostaglandin development also does very similar things to arrest the hair in its cycle in the resting phase. What about a disease like alopecia areata, where basically there's autoimmunity has then caused the hair to arrest in its telogen state? It's not growing, but it's in reinforcing a normal biological signal. Then use this kind of system and then do trials like for the tofacitinib trial that our hair research group has done, and in collaboration with a talented group, it's again a developmental biologist and translational biologists, Glenn Crispin and Justin Coe, Andrew Cristiano's group at Columbia, and Brett King at Yale. You can then develop and test these, but then develop biomarker studies using RNA-seq studies that then can actually develop something called the arson score, the alopecia areata responsiveness to JAK inhibitors, which then can nicely predict and stratify responses to responders, slow responders, and non-responders to help guide therapy. As well, you learn a lot about the biology of autoimmunity, because this list of regulators and predictors is not the usual list, and it can give you insights into disease. And lastly, just a push for stem cell biology and regenerative medicine. We're at a next in our understanding because we have this stem cell biology emerging, genome editing technology which is emerging, and tissue manufacturing from our world that can really come together and develop novel therapies for our patients. And in particular, epidermolysis bullosa, which is a debilitating disease with basically palliative therapy that's only available. Can we do something to actually correct the disease at birth and then prevent a lifetime of horrible palliation and suffering? And so this combination helped us in our lab develop what's called therapeutic reprogramming, which is in collaboration with Maurice Vernig and Al Lane's lab, to then really take the skin biopsy, reprogram to iPS cells, use gene editing technology to correct the disease, and then basically use manufacturing technology to manufacture stem sheets for grafting onto patients. And it's this technology and this group at Stanford that have really developed to then develop what's now called the Center for Definitive Curative Medicine, which we're directing at Stanford, which really, in collaboration with these other amazing folks at Stanford, to really try to translate this confluence of technologies into therapies for our patients. So I told you about autoimmunity. I've told you about tumor evolution and regenerative medicine. And none of this could really be possible without an amazing group of folks at Stanford. And I just wanted to acknowledge them here, and an amazing funding support, undying funding support from the National Institute, from NIAMS, and talented mentors such as Gene Bauer, who was chair at Stanford when I started there, current chair Paul Kavari and Al Lane, who have been instrumental in promoting my career, and also the Stanford BCC Consortium, Irvin Epstein, a longtime collaborator, and the Stanford group. So thank you very much.